Okay, so Chapter 13 is a fun one because I get to clear up a lot of misconceptions. Okay, we're going to tackle introductory virology. And we'll touch on very briefly viroids and prions. Okay, so first misconception, viruses. Okay, they go by many names. You know, we've got virus, you got virion, phage. Sometimes they're just called viral particles. The thing is, they are not cells. Okay, we established that in the first week of class. Viruses are not alive. They are non-cellular entities. And the reason we regard them as non-living, they can't reproduce on their own. Okay? Viruses have no means of reproduction. So how do they actually replicate themselves, right? Viruses can replicate. Well, they need you for that. Okay, so when you are sick with a viral infection, long story short, because we'll get into it later, they're taking over your cells, and they're turning your cells into viral factories. Okay, so they cannot reproduce without a host. They don't have the means to do it, but you do. They take advantage of the machinery in your cells. So, we call them obligate intracellular parasites. So obligate, they have to have something. Intracellular, within cells. Parasites. They parasitize your cells. Now, another misconception, people fail to realize how ridiculously simple in terms of structure viruses really are. For instance, anyone can draw the flu. Start with a hexagon. So what we have drawn here is called the capsid. It's a protein coat, a shell. So all viruses have this protein coat. The next major structure, what do you find inside of the virus? Some sort of rudimentary genome, okay, some sort of nucleic acid. So in the case of the flu, we draw in 8 segments. 1, 2, 3, 4, 5, 6, 7, 8. Each segment is a piece of RNA. That's the flu. Right? It's a hexagon with 8 pieces of RNA in it. That's it. That is how silly simple they can be. Okay, but we'll get into that more in just a second. And the last major misconception. People don't realize there are viruses of all organisms. Obviously, there's viruses of humans. We know that. There's viruses of plants. There's viruses of mushrooms. There are viruses of bacteria. Okay, and that's what we're going to focus on in this chapter. Bacteriophages. Viruses that prefer bacteria. And then even archaeans aren't safe. There's viruses of archaeans as well. So no one on this earth is safe from viruses. Okay, so getting into the very basic structure of viruses. Their anatomy is very simplistic. So let's talk about this nucleic acid core, the genome you find in viruses. This is where viruses show diversity. Okay, because viruses are classified by what you find inside of them. So for instance, there are DNA viruses and there are RNA viruses. Okay, that's not too bad, right? That's 2 categories. Let's make it worse. Some viruses will have linear pieces of DNA or RNA, and others will be circular, like a chromosome or a plasmid. So the flu, for instance, has 8 linear segments of RNA. That's its genome. Let's make it even worse. So DNA, RNA, linear, circular. Next question. Is it single-stranded or double-stranded DNA or RNA? So we're going to focus on double-stranded DNA viruses because we can relate to double-stranded DNA. But there are single-stranded DNA viruses. There are single-stranded RNA viruses, and the weirdest ones, double-stranded RNA viruses. So it's just a game of mix and match. And as we'll see at the end, there are 7 different classifications. Okay, and something I'll have recorded, the Baltimore classification system. So that's the complexity here, is what sort of genome is in there. Now, where they're all the same is that coating, that capsid. Okay, this protein coat is always made up of the same protein, capsamere. All viruses have capsids. All viruses have capsids made of capsamere. Okay, it's not particularly exciting. Now, something that is kind of interesting regarding that capsid, depending on how those proteins are organized, there are 3 major shapes. So, at the top, the most common shape, especially amongst viruses of humans, is polyhedral. Now, for whatever reason, your textbook has gone away from the more traditional term, which most other books use. Polyhedral is also called icosahedral. You may or may not know what an icosahedron is, but if you've ever seen a 20-sided dice, depends on how much D&D you've played, they look like a 20-sided dice. That is polyhedral. That's an icosahedron. And you're thinking, wait, how can they have such a geometric shape? Take a look at that TEM image at the top. Right? Looks exactly like an icosahedron. Okay? That is a polyhedral virus, and this is most animal viruses. So 20 equilateral faces. Helical viruses. Helical viruses look like filaments. They look like a helix, a spring. So with these guys, you've got this spring shape for the capsid, and technically, if I can do this without ruining the marker, the genome is coiled inside. We won't really see this. This is typical of plant viruses. Okay, classic example is the tobacco mosaic virus, which they used in every textbook until yours decided to get cute and throw in Ebola. Technically, Ebola is helical, technically, but it has an unusual version of it. It takes on what they call a shepherd's crook shape, where the ends are kind of bent over or coiled up. So, not a great example of a helical virus. Alright, now what we are going to focus on in this chapter is that strange-looking guy at the bottom. Complex viruses. And complex because of the multitude of components. So basically, with a complex virus, it has 2 major components. The head component is polyhedral. The tail sheath is helical. And then I'll talk about the little legs later on. Okay? All these parts have a purpose. So why are we looking at these guys primarily here? That's because most bacteriophages have this complex shape. And that is going to be our model virus for this chapter. Okay, so looking at external structures. Okay, the first one is optional. So viruses may be naked or enveloped. Okay, naked meaning there are no additional layers. You've got your genome, you've got your capsid shell, and then that's it. There's no other layers to be found. This is typical of most viruses, because naked viruses will punch their way into a host cell, and then they will punch their way out. They lyse the host cell. They burst it when they're done. Now, on the other hand, when you deal with certain animal viruses, you have to talk about envelopes. So, with that virus at the bottom, it's got that kind of orange shell around it. That is a membrane. Okay, it's a lipid bilayer. A phospholipid bilayer, like your plasma membranes or the cytoplasmic membranes of bacteria. But the thing is that viruses don't make membranes. Okay? They steal this from their host. So this is optional. Again, not all viruses do it, but I just want to show you kind of how this happens. All right, so see we got this polyhedral virus inside of this cell. Okay, we'll draw a couple. Why not? They need to leave the host when they're done. So in the case of enveloped viruses, they go to the cell membrane and they basically trick the cell into packaging it up. So this is a GenBio term here, forced exocytosis. So they force the cell to package the virus, and this is the result. A virus that has now acquired the membrane from the host. So that's your envelope. Okay, so as they leave, they take a little piece of the membrane with it. Okay? HIV disease. So, totally optional. This really only applies to animals that don't have cell walls. So you don't see this with bacteriophages, okay? Or with plant viruses, really. And I'll talk about the spikes here in just a second. So foreshadowing. All right, so here's another fun misconception. People fail to realize how stupid small viruses are. If a virus is to replicate within some host cell, it must be considerably smaller than that host cell. Right? It just makes sense. So, viruses range in size. Okay, 10 nanometers to over 500 nanometers. I should update that here. But take a look at some of the examples. So here's an E. coli cell. E. coli's virus is the T4 bacteriophage, right? A little complex looking dude. But that's what? Maybe 1/100th the size of the E. coli cell? Okay, sitting at about, what do we got here? 225 nanometers tall. Okay, pretty small. Over here we have a human red blood cell. I love to point this out. There's the poliovirus, sitting at 30 nanometers. Look how much smaller it is compared to that red blood cell. Okay, so viruses tend to be considerably smaller than the host cell they infect. Some do get a little bit on the big side. Ebola's a bit of a monster. There's a handful bigger than that. There's a newer class of viruses called Pandora viruses, which are almost as big as E. coli. They're really weird. Now, with this diminutive size comes limitations within the virus itself. Because some of these viruses are ridiculously small. So the question is how much DNA or RNA can you pack into there? Not much. So viruses are the image of efficiency. So let's calculate this in terms of how many genes does a virus have. And the range I have is 4 to 1,000. And 1,000 is being very generous. Okay, so for instance, the flu, if you drew this, you drew it accurately. The flu has 8 genes. 8. Think of how many hundreds of millions of people it's killed throughout history. And it's needed 8 genes to do that. Okay, polio is even more ridiculous. Polio has 6 genes. It can render a child paralyzed for life. So most viruses don't have very many genes. They don't need many to do what they do. Okay, the ones with the hundreds or thousands of genes, those are the weirdos. Okay, Ebola is unusual. There's one called the Mimi virus, which is massive and has about 1,000 genes. But with these particularly huge viruses we've recently discovered, we're starting to ask new questions like, are they really a virus, or are they something that used to be a cell? Because we don't know exactly where they fit in the grand scheme of things. We even found one Pandora virus that had ribosomes. It has no business having ribosomes. No other virus does. So, did viruses come before cells, or are they from cells? Hard question to ask. Oh, I always forget about this picture. This is just illustrating that the viruses are so tightly packed with DNA or RNA that there's actually positive pressure, right? You pop the little guy and it just explodes forth with DNA. All right, so if you remember back when we talked about bacterial cells, we spent a lot of time talking about all sorts of external structures, right? Pili, flagella, capsules, whatnot. The nice thing about viruses is outside of that possible envelope, the only other structure is spikes. Now, spikes are something you find on all viruses. All of them. It's a requirement. Because spikes are utilized for attaching to host cells. And every virus has to find a host cell. So spikes are glycoproteins, a carbohydrate sugar protein complex. And the chemical nature of a virus's spike is very much a fingerprint. Okay, they're all different because every virus recognizes different hosts. So with the flu here, to kind of conceptualize what the spikes would be like, you kind of like drawing a pin cushion. Draw some needles sticking out here. Okay, that's what the spikes would look like. Now, on the same note as the flu, I like to introduce an example of spikes that you folks already heard of, but didn't know what they actually were. So the flu actually has two spikes. H spike and N spike. Right? You hear about H1N1, H5N9, right? H number, N number. Those are the two spikes. So H stands for hemagglutinin. That is the spike they use for attaching to your red blood cells. Now, the N spike, on the other hand, this is always a hard word to say, neuraminidase. Anyways, the N spike, that is how they escape your cells. So they use this spike to trick the cell into allowing it out. Okay, so this guy's got a spike for getting in and a spike for getting out. Now you may wonder, what's up with the number? Right? H1N1, H5N9. That refers to the genetic variant of that spike. Okay, viruses undergo a lot of mutation, and they have no repair mechanisms. Okay, so spike genes, they undergo a fair amount of mutation. So we end up with all these different versions. And here's a fun thing. Different types of the flu can come together, and they can mix and match their spike genes. So this is why we worry about species jumps with like avian bird flu and swine flu. Because flu strains that are unrelated can come together, making something new. Call that genetic or antigenic drift. Okay. So we've got these spikes, right? They stick out of the virus. They use them for attaching to host cells. Next thing we have to talk about is specificity. So they use the spikes to recognize something on a specific host cell. So usually the viruses use their spikes to attach to some surface receptor or some other molecule. Okay, so for example, looking at bacteriophages. Okay, what would they look for on a bacterial cell? Well, three great examples. At the top, you have a TEM image of viruses using their spikes to attach to a pili, a sex pili. And you can see it's just totally covered. Next image down is my favorite. Here you have a bacteriophage wrapping its spikes, those little legs at the bottom, to a flagella. Because that's what they recognize, the flagellin proteins. And then the bottom one is poor E. coli with T4 phages attaching to its LPS layer. Okay, so viruses, they all recognize different receptors, different molecules on host cell surfaces. And they can get very picky about what they even recognize. Okay, the spike receptor attachment is very much a lock and key fit. To illustrate how damn picky they can get, here's another great relatable example. HIV. Okay, people with HIV get AIDS. right? Where their immune system is just totally gutted out. The reason for that is the fact that HIV only wants one cell type in your whole body, helper T cells. Right? It's got all these different tissues and cells it could pick from, but it just wants those helper T cells for some reason. Why? Because your helper T cells have the infamous CD4 receptor. It's the only cell in your body that has a CD4 receptor, and it is the receptor that HIV spikes will bind to. The infamous GP120 spikes. Okay, so spikes attached to something on the host cell surface. They tend to be fairly specific about what they want, so that leads us then to host range. How many different hosts can a given virus infect? Or how many different cell types or tissues can it infect? So what's the spectrum look like? Most viruses are ridiculously picky. Right? The T4 phage wants E. coli and only E. coli and won't touch anything else. Okay? So, HIV only wants your helper T cells. So most viruses have a very limited host range. Now, of course, there's got to be exceptions. There's got to be viruses that can infect a multitude of hosts. So, two somewhat obvious examples. Rabies. Right? The rabies virus. Think of how many little critters you are afraid of getting rabies from, right? Squirrels, raccoons, chipmunks, whatever. Okay? You can get rabies. That's because the rabies virus recognizes a protein that is on the central nervous, that is in the central nervous system of all warm-blooded mammals. Okay, so something that we all share. Therefore, the virus can infect multiple animals. Ebola is another interesting example, because Ebola actually switches things up. Ebola starts off as narrow host range. Initially, during the Ebola infection, it infects your macrophages, white blood cells. And it doesn't do much. It hitches a ride to your lymphatic system. And once it gets there, it spreads. And says, screw it, I'm going to start destroying whatever tissues I want, and just basically infects every possible cell type it can. And what happens is, your body fights back through inflammation, and often people will die from internal bleeding because of that inflammation. Okay, people who show signs and symptoms of Ebola tend to die within 2 to 3 days of showing those signs and symptoms. Okay, it burns fast. Okay, so any questions about the basic characteristics here, spikes, shapes, whatever? Okay, because now we're going to start transitioning into the replication cycles. Okay, so what is the relationship between virus and host cell? So one of two things may happen when you're infected by a virus. Okay, if you go down to the left, you may experience disease immediately. Okay, an active infection. Virus gets to work right away, making more viruses, killing your cell. Now, on the other hand, where things get interesting, is some viruses have the ability to go dormant. And they do so by integrating with your own DNA. So those incurable ones, like hepatitis, HIV, chickenpox, okay, diseases where if you get it once, you have it forever. Okay, because they have become a part of you. Okay, so as we get into replication cycles, we need to talk about what all viruses can do. Right? They all have genes. They may not have very many, but they all have certain genes. But which genes do they have in common? What traits do they have in common? So the first thing, all viruses have the genes for making a capsid. Right? They all have this shell, therefore they must have the gene for capsomere proteins. The second one's kind of a loaded statement. They will all have the genes that allow them to, in some way, be it on their own or with the host assistance, copy their genome. Okay, so the flu has the means of copying its 8 segments of RNA. And then the third thing is, the virus must have genes that allow it to get in and out of the host cell. Basically spikes. Right? All viruses have spikes, therefore they have the genes for spikes. So, comparing all known viruses This is all they have in common. This is a decent amount of stuff. But, as we'll see, there's a significant amount of subtle differences. Okay. So, like I foreshadowed before, we are going to focus on one viral example in this chapter. The good old T-even bacteriophage. And again, I have good reasons for this. Primarily, nearly all bacteriophages are double-stranded DNA viruses. Okay, so all the stuff we learned in Chapter 8 applies here as we learned it. Alright, so breaking down my complex friend here. Oh, and T-even, viruses don't often get fun names. So T-even means like T2, T4, T6, T8, and so on. Right, T plus even number. There's T-odd phages too, right? T1, 3, 5, whatever. Alright, so at the top of my evil little moonlander here, I have the capsid. And as you can see in the cross-section, it is polyhedral and packed with DNA. Now attached to that, we have the tail sheath. This is very much like a spring. Okay, so treat it like it's a spring. Alright, so we got this helical tail sheath, and then that ends in a base plate, which we're not going to mention. But attached to the base plate, we've got the tail fibers, which are spikes. Spikes don't necessarily have to stick out of the capsid. In this case, they're the little legs that it lands on. And then the pins, I'm going to come back to these little fang teeth-looking things here in just a minute. Okay, but the point I want to make here is that in the pictures, I mean, it looks plastic, right? Or maybe metallic. You gotta keep in mind, this thing's organic from top to bottom. Okay, it's all protein and DNA. Alright, so what we have here, it looks like a big piece of poop, is E. coli. And poor E. coli is currently under siege by a multitude of T4 bacteriophages. So, I'm going to go back and forth with this picture. Okay, so we've seen what all viruses have in common. Let's talk about bacteriophages, okay? What kind of sets them apart? Alright, so, first thing. For these bacteriophages, once they attach to the host cell, they inject their DNA into the bacteria. Like evil little syringes. Second thing. Once they've injected their DNA into the host cell, what remains on the outside is an empty husk, a shell. So you can see all the little blue guys there. Those are probably empty, like an insect molting. Okay, so they're spent. Once that DNA gets into the host cell, a lot of little things happen. But, long story short, it takes over the host cell, forces the host cell to copy the DNA, and it forces the host cell to transcribe and translate the viral genes. Okay, so the host is forced to do everything here. And then the last thing is, we're going to see there's two major replication cycles, as I kind of mentioned earlier. The lytic and lysogenic. Alright, so, yes, or Doreen.
So what happens to the empty ones, the empty shells that are left outside do they die off? So once the virus gets into the host cell,
they're just discarded. They'll probably just break down over time. Just like an insect molting, right? What happens to that cicada shell stuck to the side of a tree? You know, just kind of falls off and breaks down over time. Alright, so this is something that kind of applies to all viruses. Okay. Oh, sorry, Charles, go ahead.
So if the tail fiber has spikes, how does the capsid use the spikes?
We'll see. Yeah, we will see, I promise. Alright, so two major replication cycles here, and this is for all viruses. Okay? Some viruses are lytic, meaning when they infect the host cell, they immediately get to work, making more viruses and killing the host cell. Okay? They will destroy the host in short order, an active infection. Now, lysogenic phages are a little confusing. Okay, because for one, they can do everything a lytic phage can. They can go through this lytic cycle we're going to talk about. But what makes them lysogenic is that they have an added bonus. They have the ability to go dormant by hiding in the host DNA. And it's going to be forced recombination. Right? They will force their DNA into the host's DNA. And then it becomes a mutation. And it can pass down through the generations. Okay. So, we're going to look at the lytic cycle first. It's really not terrible. We got 5 major steps. Okay? Attachment, penetration, biosynthesis. I got to fix this. Assembly, or another term is maturation. And then release. Okay. So let's go through the overview first. So at the top, this is attachment. Right? The virus has used those tail fibers, its spikes, to attach to the surface of E. coli. What next? Like I said, this guy's like an evil little syringe. Right? That tail sheath is very much a spring. So it takes that capsid and smashes down on the base plate, injecting its DNA into the host cell. Right? So you can see here, there's that DNA being injected. Third step, and this is the most complicated for all viruses, biosynthesis. Okay. Virus takes over the host cell, forces the host to make more viral components. Step 4, some books call it maturation, some call it assembly. Assembly makes more sense to me. But like evil little Legos, the host cell is forced to assemble mature viruses. And then the 5th step is release, where the obvious happens. The virus busts out of the host cell, they go out, they find new host cells, and it begins again. Attach, penetrate, biosynthesis, maturation, release. Alright, let's talk a little bit about what's happening in these steps. So, looking at T-even phages here. So, in attachment, we've discussed this already, right? Spikes will bind to some receptor or protein on the surface of the host cell. So for the T4 phage, as to my understanding, it binds to the LPS layer of E. coli. Now the penetration step is very clever. Okay, this is where bacteriophages are very different from animal viruses. Because for a bacteriophage, okay, imagine you've landed on the surface of E. coli. You got to deal with one big barrier, the cell wall. Right? When I talked about peptidoglycan in the past, I said it's like stacking and welding chain link fence, and I meant it. So you're a virus, you got to get through all those layers of chain link fence. How do you get through that peptidoglycan? You're going to use a ridiculously clever enzyme, lysozyme. Jump back here real quick. Sorry to make you flip if you do. So those pins, the little fangs, that's lysozyme. It is an enzyme we've mentioned once way back in chapter 3. No, 4. Lysozyme is an enzyme that is found pretty commonly in nature. Okay? We are producing it now as humans in our tears, saliva, and mucus. It is an enzyme that very conveniently breaks down peptidoglycan. So, our virus here, and this is relating to ice fishing, has a built-in auger or drill. Right? In ice fishing, they have to auger through the ice to make a hole. Okay? Here, the virus is going to use lysozyme to drill a hole in the peptidoglycan. And once it's done that, capsid smashes down, injecting the DNA. Again, like an evil little syringe. Alright, now, the biosynthesis step. This is the hardest part of every virology chapter. Because no two viral families go about this step the same way. Okay, so I'm going to try to water it down best I can. So at this point, imagine the viral DNA is floating around the inside of the bacteria. What comes next? transcription and translation. The host RNA polymerase, its machinery, is tricked into expressing those viral genes. Okay, so you're going to make viral proteins. Now, something interesting about viral gene expression is that it's chronological. When the genes are expressed is timed in most cases. So we'll often classify viral genes as being early, middle, or late. And early, middle, or late just means when is the gene expressed during an infection? At the beginning, in the middle, or at the end? So why would they be timed, right? There's good reasons. It's when they're needed. So a great example of an early gene. One that produces nucleases. Okay, nucleases are enzymes that chew up DNA. To defend itself from the host, the virus will chew up the host chromosome. Right? Chew into pieces so the host can't defend itself. Okay, you need those nucleases to do that right away. Now, what about a late gene? Right? What enzyme or protein would you need towards the end? How about lysozyme? Right? Lysozyme may have got you into the cell, but if you make it too early, it's probably going to break down the host cell wall prematurely. So you're going to want to save that lysozyme for the end of infection when you need to cut your way out. So there is logical reasons for why they express their genes at different times. Doreen.
So you say lysozyme gets you in and gets you out.
It does. And that's what we're going to see with the release step. Lysozyme comes back. But, in the end, okay, two major things happen here. One, the host cell is forced to copy the viral DNA. Right? You need to make more copies of that viral genome. The other thing, the host machinery is forced to, as we just said, express those viral genes. Make viral proteins like spikes or capsid proteins. Right? We need to fill the cell with all the little pieces we need to make more viruses. And where the raw material comes from? From the broken down host cell. All right, now, maturation. Like I said, this step's also called assembly, because now the host cell is forced to put the viruses together. Take all those little components we're making, put them into mature viruses, and this is something else that is done in a precise order. Okay, so for instance, I want to make a mess of the slide here. The DNA is carefully woven into the capsid. Very carefully. The tail sheath is also assembled separately. Once those are assembled, they are combined, and then the last thing that gets attached are the pins and the tail fibers. So again, done in a highly chronological order. Fifth step is release. So like Doreen just mentioned, lysozyme not only gets this virus into the cell, but lysozyme is how they carve their way out. So they use those pins to thin out the cell wall until it bursts. The viruses get out, and then the cycle begins again. Find a new host cell, attach, penetrate, biosynthesis, maturation, release. Doreen.
Again, I want to ask. So in this part, you say the host DNA and cell components become the building blocks for the phage.
Right.
Components, right?
Yep. The virus not only breaks down the chromosome, but it'll break down the host proteins as well. Because we want to make a soup of
So they
nucleotides.
become building blocks.
Yep.
For the
Yep.
virus.
Yep. So they just break it down into nucleotides and amino acids. So they can use that to make their own components. Alright, so, fun facts. Certain things are very calculable here. People wonder, how many viruses can you make per cell? Well, for bacteriophages, they'll make about 200 mature viruses per bacterial cell. How long does it take? Pretty fast. For T4, it's about 20 to 40 minutes. So for our lecture, that's almost 3 generations for this virus. And you can actually see this happen. So we mentioned plaque assays in the last chapter, okay, where we use viruses to identify strains. Here, we've got 3 plates of E. coli. E. coli is being infected by 3 different viruses here. T2, T4, and Lambda. Now you'll notice there's clearings on all 3 points. The clearings are what you call plaques. Basically, the clearing indicates the virus has destroyed the bacteria, annihilated them. But notice how the plaques are different sizes? Different viruses replicate at different speeds. So, T2's the slowest, T4 is a little faster, and Lambda is really, really fast. Okay, so you can see this happen. Alright, so there is an animation in here for you, buried away, in case you want to watch this in action. But for all viruses, they go through these 5 steps. The differences lie in how they go about each step. And if you ever get a chance to take a virology class, jump on it. Because it's an interesting class to take, it's a hard one to teach. Because each chapter is a different family of viruses. And each chapter is reliving the same story over and over and over again, where it just asks, how do they go about these 5 steps? Chapter after chapter after chapter. Okay. See how far we get into this next one. Any questions about those lytic phages? Alright, so what we're going to look at next, or at least in part, are lysogenic or temperate phages. Okay, these are viruses that can, again, either go through the lytic cycle, or they can go dormant. They can go through this lysogenic cycle. Okay, so what is transpiring here? Well, it all starts off the same. You get attachment, you get penetration, but for lysogenic viruses, they have to make a decision. Okay, so this is phage lambda. It's got a circular genome. It has to decide, well, am I just going to go through the lytic cycle, killing the host right away, or am I going to go dormant? Now, when I say dormant, I mean the virus will integrate itself into the host chromosome. Okay, so how does it do that? Through something called site-specific recombination. Okay, kind of like we talked about recombination in Chapter 9, it does put itself into the chromosome. It recombines itself. Now, some terminology here. There is a term for a virus that has gone dormant. We call it a prophage. So if you've ever had chickenpox, you know someone with chickenpox, they harbor the prophage for that virus in their peripheral nerve cells. It's a part of their DNA now. Now, what do you call the cells that harbor the prophage? Some backwards terminology here. We call it a lysogen. You're like, well, wait, shouldn't that be what you call the virus? No, the lysogen is what you call the cell that harbors a prophage. So, in the case of chickenpox, okay, the prophage is in your peripheral nerves, therefore your peripheral nerve cells are lysogens. Once the virus is established, bad news, it stays there for some time. It gets passed on to future generations of that particular cell. But the thing is, this is merely hitting the pause button. Because eventually they're going to finish that show. The virus will reactivate by removing itself from the chromosome, and then it makes the decision, okay, time to go through the lytic cycle. The inevitable always occurs. The virus reactivates, makes more of itself, kills the host cell. Okay, so in the case of chickenpox, that is when you experience shingles later in life. The reactivation of that same virus from those peripheral nerves. Okay, and I drag my hand across my torso because that's a typical peripheral nerve that the virus would reactivate from. Okay, so now we have another fun question to ask. How does this virus get to make a decision? Right? How does it know, I'm going to do the lytic cycle, or no, I'm going to go dormant? It's not super well understood. But one trend we have seen, one correlation, is the current growth state of the host cell. So with bacteria, remember the growth curve, right? Lag, log, stationary, death. If the host cell is growing slowly, say stationary phase, the virus is more likely to become a prophage. Okay? Because recombination takes time. Host cell's growing slowly, it can take the time to put itself into the chromosome. Now, if the host cell is growing rapidly, think log phase. Okay, for one, there's no time to become a prophage, but two, you got more host cells to have fun with. So more typically, the virus would go through the lytic cycle. Okay, so host cell growing slowly, odds are it become a prophage. Host cell growing rapidly, odds are it goes through the lytic cycle, killing the host cell. Okay, so this is a diagram that seems a little complicated, kind of is, but it kills me that the textbooks are starting to exclude it. But this is how the virus puts itself into the chromosome. This is a very, very simple version of site-specific recombination. Okay, so let me show you. All right, so the pink circle at the top, that is the chromosome, the genome of phage lambda, a virus known to infect E. coli. Okay, it's got a circular chromosome. Now, on its chromosome, it has these P sites, P and P prime. It's a little sequence of DNA. When phage lambda infects E. coli, it looks for E. coli's chromosome because on E. coli's chromosome, you've got these sequences called B and B prime. Whoops. Those sequences are complementary. Okay, meaning they match. Okay, evolution's a funny thing. So when the lambda DNA gets into E. coli, these sites align, and then, leaving out a lot of details here, you can see that the virus loops in. Okay, so it kind of crosses over, loops in, and at the bottom, you can see it has shoved itself into the chromosome. It's an established prophase, and it's going to stay there until it decides to leave. So, enzymes always get fun names. The enzyme the virus produces to put itself into the chromosome is called integrase. Why not? Now, when a virus wants to leave, it does it all in reverse. Okay? Loops back out, crosses over those sites, pops out. That enzyme's called excisase, right? Because that's going to excise it. Anyways. Oh, I got a couple minutes for this. So my recording I'm going to post later already talks about this, so you may hear it twice. Or you can skip it. Another fun thing to think about with prophages. Okay, this is a virus that's gone dormant. It's a part of your DNA. How dormant is it? Right, it's got genes. Are all the genes turned off? Typically, no. Some of them are left on. And those that are left on alter your phenotype. They alter your behavior, your traits. Subtly, but they do. So, this is what we call phage conversion. The prophage alters the phenotype of the host cell. Okay, so how does it do that? Well, three major examples. The first one, and this is the most common example. Prophages will produce proteins to prevent, here's your word of the day, superinfection. Meaning, preventing related viruses from infecting that cell. Now, it makes sense when you think about it, okay? Imagine, you're a prophage. You're hiding out in some bacterial cell. You don't want a related virus to show up and kill that host cell you're hiding in. So you're going to mask yourself. You're going to mask that host cell. Okay, so superinfection only applies to related viruses. Okay, it's immunity to related viruses. It does not apply to unrelated viruses. So with phage lambda, right, it can prevent other lambda viruses from getting at E. coli, but it can't stop, say, the T4 phage we saw before. The second one's a scary one. Many prophages come with genes for producing pathogenic toxins. This is a shockingly long list of bacterial pathogens that are only pathogenic because of toxins from prophages. Diphtheria, botulism, I'll skip that one, scarlet fever, food poisoning, cholera. All of these are the result of bacteria that happen to harbor a prophage that just happens to have a gene for a nasty toxin. So going back to that one I skipped. Good old E. coli 0157H7. Okay, normally E. coli is harmless. But 0157 is special because, as we mentioned in an earlier chapter, it produces the Shiga toxin. So how does it do that? 0157 harbors a prophage that has the Shiga toxin gene. That's the only reason why it's pathogenic. It just happens to harbor a prophage, allowing it to be pathogenic. And then the third example, the prophages will produce proteins that maintain their self-latent state. Why that's phage conversion? Because sometimes they require the assistance of the host cell to do this. So sometimes the host has to help to repress the viral genes. Okay, so that's phage conversion, right? The prophage will produce proteins that alter the host behavior. And we'll do this one real quick and I'll be quiet because that will be a good stopping point. Alright, so the last question to ask with these lysogenic phages. Okay, how do they choose to reactivate? Right, at some point the inevitable occurs. The virus leaves the genome and finishes out the lytic cycle. Again, not well understood. But this is what we do know. We do know that host cell stress or damage can cause the virus to reactivate. Okay, right, with chickenpox, once you get over it, you're not really worried about having shingles again anytime soon. Right, you're worried about getting shingles when you're 65 and older, when your body's becoming weaker and can't maintain the repression of that virus. So the likelihood of it reactivating goes up. But otherwise, it can be totally spontaneous. Okay, while most old people do get shingles, there are people in their 20s that can get it. Just because the virus said, screw it, I'm going to reactivate today. Okay, so it all depends. But there's a multitude of viruses that do this. Okay, any related to HIV, any in the herpes virus family, chickenpox, which is technically in the herpes virus family, amongst others. Okay, so again, just hitting the big point here. Lysogenic phages can do everything a lytic phage can. Right? They can either go through that lytic cycle, or they can become a prophage for some time until the inevitable. They reactivate and finish out that lytic cycle. Again, hidden animations, please watch them if it helps.